Welcome back, everyone. I hope you had a chance to enjoy your lunch and recharge. Just a few reminders. This workshop is being recorded and will be posted onto FDA's website following the workshop, along with all of the presentations. We are now ready to move on to session 2 of our workshop, Novel Bioequivalence Study Design Recommendations. As a reminder, you may submit your questions through the MS Teams Q&A function, or you can send us an email at orsevents@fda.hhs.gov with the subject line AGDD 2025. We just ask that you please indicate who the question is for so that we may route it appropriately. Now, it is my pleasure to introduce Dr. Angelique Bissold, a senior pharmacologist in the Division of Bioequivalence 2 within the Office of Generic Drugs at the FDA. Dr. Bissold's primary responsibility is the assessment of bioequivalence studies submitted as part of abbreviated new drug applications. Prior to joining the FDA, she completed her PhD in pharmaceutical sciences at the University of Maryland School of Pharmacy and postdoctoral research at the Johns Hopkins School of Public Health. Today, Dr. Bissold will be introducing our session 2 presenters. Please join me in welcoming Dr. Angelique Bissold.
Thank you, Maria, for that kind introduction. Hello, everyone. As you've heard, my name is Dr. Angelique Bissold, and I am the lead for session 2, along with Dr. Andrew Babiskin, who is a lead pharmacokineticist in the Division of Quantitative Methods and Modeling within the Office of Research and Standards. Together, we'd like to welcome you to session 2, Novel Bioequivalence Study Design Recommendations. In this session, you'll hear more about challenges in establishing bioequivalence for a variety of drug products and the innovative research and methods that have been used to overcome these challenges. We have 5 fantastic talks lined up for you today from 8 speakers, all within the Office of Generic Drugs. You'll first hear 2 talks about challenges in Biopharmaceutics Classification System, or BCS waivers. The first talk will provide an overview of BCS waiver options and product-specific guidances and provide updates on the FDA's initiatives, regulatory efforts, and research outcomes to promote and advance BCS-based biowaiver options in generic drug development and approval. This presentation will be given by Commander Yi Zhang, Senior Scientific Advisor in the Division of Therapeutic Performance 2 within the Office of Research and Standards. Commander Yang has a wide range of expertise including in the development of product-specific guidances, responding to public inquiries such as controlled correspondences and citizens' petitions, managing pre-ANDA meetings, addressing internal consults, and leading multiple research projects to establish novel bioequivalence approaches. Next, we'll hear more specifically about addressing degradation challenges in BCS Class III based biowaivers through physiologically based pharmacokinetic modeling from 2 presenters within OGD. Dr. Usha Katragada is a senior pharmacologist in the Division of Bioequivalence 3 within the Office of Bioequivalence, where she's primarily responsible for assessing the bioequivalence of various dosage forms of generic drugs. She also has expertise in transdermal delivery dosage forms and nanoformulation development. You'll then hear from Dr. Fang Wu, a senior pharmacologist in the Division of Quantitative Methods and Modeling within the Office of Research and Standards, who is responsible for using modeling and simulation tools for reviewing pre-ANDA meeting packages, ANDA consults, and controlled correspondences. We'll then switch gears to hear more about overcoming challenges in carryover seen in long half-life drugs with insufficient washout. This will also be a joint presentation from 2 speakers within OGD. First, you'll hear from Dr. Min-Zhu Chung, a pharmacokineticist in the Division of Bioequivalence 3 within the Office of Bioequivalence, who works on the evaluation of generic drug applications, in addition to work dedicated to safety evaluation of inactive ingredients for pediatric populations. Next, you'll hear from Dr. Denise Osden, a pharmacologist in the Division of Quantitative Methods and Modeling within the Office of Research and Standards. She is responsible for reviewing and responding to controlled correspondences, consults, and pre-ANDA meeting requests for generic drug-related submissions and developing product-specific guidances. You'll then hear updates on specific considerations pertaining to subject safety in pharmacokinetic bioequivalence studies from 2 speakers within the Division of Therapeutic Performance 2 within the Office of Research and Standards. Dr. Karen Lee is a pharmacologist whose current work primarily focuses on the protection of human subjects in bioequivalence studies by providing guidance and addressing safety inquiries related to product-specific guidance development and pre-application support. Dr. Duyin Nguyen is a pharmacologist who has expertise in clinical safety assessments, contributing to the development of product-specific guidances and addressing public inquiries through controlled correspondences, pre-ANDAs, and docket comments. Her regulatory research focuses on various aspects of subject safety and bioequivalence studies. Finally, we'll learn more about the alternative approaches for establishing pharmacokinetic bioequivalence when the reference-listed drug or reference standard is unavailable. This talk will be given by Dr. Yuqing Gong, who is a senior pharmacologist in the Division of Quantitative Methods of Modeling within the Office of Research and Standards. Her research focuses on utilizing quantitative tools such as population pharmacokinetics, modeling, and simulations to address specific questions related to generic drug development and/or regulatory decision-making. With that, I would like to welcome Commander Zhang to start us off with session 2.
Thank you very much. Good afternoon, everyone. Really appreciate that nice introduction of all our speakers in this afternoon session. And let's kick off this session with my presentation entitled Biopharmaceutics Classification System Waiver Option in Product-Specific Guidance. Disclaimer, the presentation reflects my own views and should not be construed to represent FDA's views or policies. There are two objectives for my today's presentation. One is to describe the current FDA's standard practice, regulatory process, and the scientific framework in recommending BCS-based Biowaiver as an alternative B approach within PSGs. The other is to present FDA's initiatives, regulatory efforts, and research outcomes to promote and advance BCS-based biowaiver options in generic drug development and approval. Here, I'm pointing to the potential challenges by applying the BCS-based biowaiver. I will have more details in the following. The ICH-M9 guidance set a good foundation for BCS-based biowaiver. This guidance was published in May 2021. I have provided a link here for you to further refer. This guidance is intended to provide the BCS-based biowaiver to immediate release solid oral dosage forms like tab, capsule, and suspensions, which are intended to deliver the drug to the systemic circulation. There are some exceptions for this guidance. One I want to point out to bring your attention is the narrow therapeutic drug products are excluded, as well as some other dosage forms like sublingual, buccal tablets. They are excluded from this waiver eligibility. The scientific foundation for the BCS-based biowaiver is established on the scientific framework for BCS classification based upon aqueous solubility as well as interstitial permeability of the drug substances. Let me walk through you all to refresh ourselves about the BCS classification criteria. This table showed there are 4 BCS classes of drug substances. 4, 1, and 3 have high solubility, 2 and 4 have low solubility. For 1 and 2, they have high permeability, while 3 and 4 have low permeability. So the high solubility is defined as the highest single therapeutic dose is completely soluble in 250 milliliter or less of aqueous media over the pH range from 1.2 to 6.8 at 37 Celsius degree. And the high permeability can be concluded by the following: when the absolute bioavailability is not less than 85%, or if not less than 85% of the administered dose is recovered in the urine as unchanged parent drug, or as the sum of the parent drug, including phase 1 oxidative and phase 2 conjugative metabolites. Last, I want to remind the audience that, per this M9 guidance, the significant degradation of more than 10% of a drug substances will prelude its BCS high permeability classification. We're all aware the benefits of the BCS-based Biowaiver, which is a less burden alternative B approach for IR solid oral dosage products. The overall is to reduce both the costs and unnecessary human drug exposure for developing the generic drugs, especially to facilitate the generic oncologic drug development by elevating the need to conduct B studies in patients commonly often challenged with the difficulties in patient recruitment or healthy subjects which may pose potential safety concerns, as well as to expedite the process to promote generic drug development and approval for complex drug products, such as containing complex API, I will showcase later, by avoiding the challenges in conducting comparative clinical endpoints B studies, as well as the challenges in accurate measurement of PK in vivo. Then you may wondering, when does a PSG recommend a BCS-based Biowaiver option? In general, PSG includes a BCS-based Biowaiver recommendation after the agency has determined the drug as either BCS Class 1 or 3 drugs. As of up to date, the BCS-based Biowaiver option has been recommended for in the PSGs for 59 BCS Class 1 drugs and 10 BCS Class 3 drugs. Here, I just want to show you the most recent PSG example for hydroxyurea tablet, which was just published early this month. So you can see in the recommended study row, 2 options are recommended. One is BCS Class 1 based Biowaiver. Second is the in vivo B study with PK endpoints. You can see some most recent up-to-date standard language from the agency regarding the BCS-based Biowaiver in this PSG example. The second example I want to show is about the oncology drug, capecitabine tablets. It's not just an oncology drug. It has cytotoxicity as well. So current PSG, we do recommend BCS Class 1 based Biowaiver option, as well as the in vivo B study with PK endpoints option. One good news is the agency has approved the one ANDA for this drug product through this BCS1 based Biowaiver approach. This is another PSG example I want to share with you about the complex API for pentosome polysulfate sodium capsules. The current PSG does recommend 2 options as well. One is BCS3 based Biowaiver option. The other is in vivo B study with clinical endpoints. You probably see the huge benefit shown here. So basically, we can through this BCS3 based Biowaiver approach, we can actually avoid the less sensitive, very costly clinical endpoint study here. I have shown you very positive cases of our PSG examples reflecting either BCS Class 3 or BCS Class 1 based Biowaiver. You might wondering, is the BCS based Biowaiver a possible BE approach, even it is not recommended in the PSG? The answer for me is absolutely yes. There are hundreds of drugs that could be eligible for either BCS Class 1 or 3. So PSGs just currently represent the conclusion of FDA assessment, but you definitely can be the first applicant to consider for BCS-based Biowaiver option to support a specific generic drug approval. I do have a positive case here in the following. For recycling mesylate tablet, that's the first BCS3 drug product determined and classified by the agency based on only ANDA data. So current PSG does reflect two options. One is BCS3 based Biowaiver option. The other is in vivo B study with PK endpoints. Another positive feedback to the audience is we do have multiple ANDAs approved for this drug product through the BCS3 based Biowaiver approach. Now I want to share some identification strategies for applying the BCS-based Biowaiver candidates. So you're probably wondering what IR products might be potential candidates for BCS-based Biowaiver, even when the PSG does not recommend it as an option. So basically, all the drugs, if they have high solubilities, they may be eligible for the BCS-based Biowaiver. There are many factors for you to consider before you take this approach. First, identify your interested drug substances and figure out if it is a highly soluble drug substance. As the solubility is not difficult to measure at all, so it shouldn't be a hurdle for you to take this BCS-based Biowaiver approach for onward. For BCS3, please keep in mind, check your test product and the ROD for very rapid dissolution testing, and as well as compare your proposed test formulation with the ROD, because we do have a Q1 same and Q2 similarity criteria based on M9 guidance. If you're not sure whether it is BCS1 or BCS1 drug, at least a BCS3 based Biowaiver can be requested. There's some advantage here because there is no permeability data you need to collect or generate it. Well, nothing is for all good. They always have pros and cons. So the challenge or disadvantages here is you have to meet the formulation criteria, which is Q1 same and Q2 similarity, and the very rapid dissolution. Here, I just want to let you know, in my part 2 presentation, I will show some agencies' effort and research, also the outcome, help dealing with those challenges. So here come my part 1 conclusions slide. I want to conclude it with how to how to request the BCS-based Biowaiver to the agency. First, you may submit control correspondence to the agency. However, you do request if your proposed product is eligible for BCS-based Biowaiver with supporting documentations and information. Please don't just ask if your proposed formulation is Q1 the same, Q2 are similar to the ROD. Second, you may submit pre-end meeting request for BCS-based Biowaiver with supporting information and documentation. Last but not least, you can go ahead and submit ANDA for BCS-based Biowaiver with appropriate supportive data regarding high solubility and high permeability for BCS Class 1 drug, while high solubility, very rapid dissolution, and the Q1 the same, Q2 similar formulation to the ROD for the BCS3 drug product. Now I'm going to switch my gear to share or present the FDA research effort and the initiative regarding to dealing with the challenges I mentioned earlier. Here's my first case. So what can you provide for the totality of the evidence to justify acceptance of drug substance degradation exceeding 10% as per M9 guidance? The case here is about cladribine tablet. Currently, the PSG recommend BCS3 based Biowaiver option. The agency used the PBPK modeling to justify the eligibility of the BCS3 based Biowaiver for cladribine, which has greater than 10% degradation. And the more details I will save for my next presenters, Dr. Usha Katragada and Dr. Fang Wu. The second case I want to present is our collaborative internal FDA research on exploring the formulation flexibilities for BCS3 based Biowaiver. We noticed some deviation from the criteria of Q1 the same and Q2 similar formulation to the ROD for the putative BCS3 drug product as per M9 guidance. Our research outcome on the formulation assessment has been published in Pharmacokinetics in 2023. I put the title of the publication and also resource here. If you are interested, you're welcome to explore more. In today's presentation, I really want to highlight the formulation survey results from our publication. From this pie chart, you can see the orange plus gray is about 70% of the approved ANDA products, which have been demonstrated BE to the corresponding ROD. They don't meet the formulation criteria as per current M9 guidance. The blue and dark orange add together is about 30%, like 6% Q1, Q2 the same, and then Q1 24% is Q1 same and Q2 similar. This is a very small portion of our formulation survey. By the way, just this research based on 133 approved generic drug products, and which we identify as putative BCS3 drug. So you can see majority of the approved drug product, they still demonstrate BE even though they don't meet the formulation criteria. I would like to share some research findings based on our formulation assessment shown earlier. So the commonly used excipients collected from 133 approved ANDAs may not affect intestinal absorption of 16 investigated putative BCS Class III drugs. Second, the rate and extent of absorption of those drugs appear to be more dependent on the biopharmaceutic and the physiologic properties of the BCS Class III drug substance, and less or not depend upon their formulations with different commonly used excipients and excipient classes. Last but not least, this study may help to explore more flexibility for BCS3 based Biowaiver recommendation regarding the current formulation criteria, Q1 the sameness and the Q2 similarity as per M9 guidance, particularly for the drug substances with moderate permeability. The last case I'm representing here is from the collaborative research with our sister division, DQMM in ORS. So this research is exploring how to use the PBPK modeling to explore the flexibility for dissolution criteria. So based on our internal collaborative research with OTR in the agency, we identified this drug product It's a non-Q1, Q2 formulation. They didn't meet the very rapid dissolving criteria. That means they didn't meet not less than 85% in 15 minutes as per M9 guidance. However, we do have, it is approved ANDA, so it has demonstrated in vivo BE to the corresponding ROD. So our DQMM colleagues developed and validated PBPK modeling for this drug substance. And they used this PBPK model to conduct virtual BE simulations to establish the BE dissolution safe space for this product. And this research outcome has been presented at ASCPT this year. If you look at the graph on the right, so you can see even for the dissolution, they do have flex spaces doesn't allow us still have the in vivo BE. So that's another good case from FDA research. Explore the potential flexibility as per our current M9 guidance. Then this is my summary slide. I want to really focus on 3 areas. First, FDA current implementation status. We have established a comprehensive framework supporting 6 to 9 PSGs with BCS-based Biowaiver options, demonstrating systemic evaluation and approval processes for generic drug application from the agency perspective. Second, FDA innovative research. We have advanced scientific understanding through product risk mitigation studies and the PBPK modeling approach to support the potential expansion of BCS-based Biowaiver eligibility, which is beyond the current M9 guidance criteria. And the last but not least, about our regulatory innovation. So we have actively developed a scientific framework and approaches to provide BCS-based Biowaiver option to the PSD and enabling faster generic access while we still maintaining our scientific rigor. Thank you very much. I really appreciate it. And the next slides. Sorry, just give me a second. The pop-up restart. Okay. Let's see. Can someone help me? I'm sorry. Because I do want to show my appreciation to all of my colleagues. Okay, here come my last slides. I really want to give my heartfelt appreciations to all my ORS colleagues and Dr. Ping Ren, Dr. Wenchen Yang, Dr. Fang Wu, and Dr. Eridan Pao, Dr. Qi, Dr. Dongmei Lu, Dr. MJ Kim, Dr. Lei Zhang, and Dr. Rob Leinberg. Thank you very much. I will back for the panel discussion for any questions. And now I'm introducing our next speaker, Dr. Wusha.
Thank you. Good afternoon, everyone. I know it's a tough time slot. Let's beat the food coma together. A small tip, chew a gum or imagine this talk as a strong coffee. Today, I'm co-presenting with my wonderful colleague, Dr. Fang Wu, and our topic sorry. Just give us a moment, please.
There you go.
Thank you. So we are presenting together on addressing degradation challenges in BCS Class III Biowaiver applications through PBPK modeling, that is physiologically based pharmacokinetic modeling. This presentation represents a paradigm shift in how we approach complex bioequivalence challenges, moving beyond rigid guidelines adherence to science-based and risk-assessed decision-making. So the learning objectives for this presentation is, first, we will browse through the Biopharmaceutics Classification System, BCS, general consideration for BCS 3 Biowaiver. And second, dive into case study to evaluate bioequivalence based on the totality of the evidence. Finally, we will explore how PBPK modeling demonstrating its power to bridge the gap between in vitro observations and in vivo clinical relevance. Before we proceed, I want to emphasize that this presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. The foundation of my discussion rests on ICH M9 guidance for BCS-based Biowaiver published in year 2021. BCS3 drug substance classified as BCS3 based on its characteristics high solubility and low permeability, which means it dissolves rapidly and limited ability to cross the biological membrane. And the three key elements for the criteria for the eligibility of BCS3 biowaiver is high solubility, formulation similarity, rapid in vitro dissolution release. Coming to the specific criteria required for BCS3 based biowaiver is drug substance should be classified as highly soluble if the highest single therapeutic dose is completely soluble in 250 ml or less of aqueous media over the pH range of 1.2 to 6.8 at 37 degrees Celsius. It looks very straightforward. However, the challenging comes to use a validated method to determine concentration of drug substance. And the drug substance stability should be demonstrated with less than 10% degradation. Excipients qualitatively same and quantitatively similar as the reference listed drug as defined in the M9 guidance. The other criteria is it should demonstrate very rapid dissolution, that is 85% dissolution within 15 minutes at pH 1.2, 4.5, and 6.8. Let's dive into case study for tablets with acid instability challenges. Reference listed drug is Mavenclad cladribine tablets available in single strength 10 milligrams, approved in year 2019, indicated for relapsing forms of multiple sclerosis. Maximum daily dose is 20 milligram per day, which indicates this drug product is potent and requires careful evaluation of bioequivalence. From RLD label, the critical information about this drug product is cladribine is acid labile. So it undergoes hydrolysis and forms 2-chloroadenine, and it is stable in neutral pH and slightly basic conditions. So as it is acid labile, the innovator combined cladribine with a complex 2-hydroxypropyl beta cyclodextrin to form more stable drug substance, increasing its solubility and stability over time. Understanding cladribine metabolism is a critical aspect in this case study. So from the metabolic pathway found in FDA clinical pharmacology review, it shows that the major degradant is 2-chloroadenine, which is about 7.8% recovered in urine after oral administration of cladribine tablets. And another interesting thing about this product specific guidance for cladribine tablets is before there is only one recommendation is recommended in PSG, that is one in vivo bioequivalence study with pharmacokinetic endpoints in patients with relapsing forms of multiple sclerosis, which is challenging and very expensive. However, now the PSG guidance has been revised to include an option of BCS Class III based biowaiver, which demonstrates there is an advancement in regulatory science and also patients' access to increased access of generic drugs to the patients. Let's turn into the evaluation of the totality of evidence in a case study. In general, for this drug product, Q1, Q2 formulation similarity can be achieved. However, the major challenge occurs in the solubility with greater than 10% degradation at pH 1.1 to 6.8 for API and pH 1.1 to 1.8 for complex. So per guidance, the solubility should be demonstrated with less than 10% degradation. So that's a problem if it is more than 10%. And for this drug product in general, due to this degradation, the probability of less than 85% dissolution is seen at pH 1.1, that is 0.1 normal HCl within 15 minutes. So again, per guidance, it should be very rapid, more than 85% within 15 minutes. So we developed a comprehensive scientific approach to address each concern. The first concern is a relationship between API and API complex. Before absorption, cladribine separates, this information is from the literature. Before absorption, cladribine separates from cyclodextrin, making the stability and solubility of both the API and API complex potentially important factors. So you might be wondering, per guidance, the solubility of API is required. However, in this case study, both the API and API complex solubility data is a critical information. Second, and another challenging part is instability at pH 1.1 to 2. Per RLD label, co-administration of pantoprazole, which is a PP proton inhibitor, PIP inhibitor, with cladribine tablets shows no effect on the rate and extent of the absorption. So with the pantoprazole, actually the gastric pH kind of increases to approximately 3.5, which shows that the bioavailability of the cladribine tablets is no difference when compared with the PPI and without PPI, which tells that the observation of in vitro degradation has no relevance in the clinical observation. Coming to the dissolution concern, considering dissolution data at extending beyond pH 1.2 to pH 1.6 using fasted state simulated gastric fluid, in addition to data pH 1.2, 4.5, and 6.8 is a good idea, as BCS3 waiver is only is applied to when the drug is administered to water, and the pH of the gastric pH with water changes, and it is more approximately around 1.6. Coming to the degradants, from the publicly available data, the degradants has no clinical relevance. And coming to the solubility study time frame, the Tmax range for this drug product is 0.5 to 1.5 hours. So 2 hours may be a suitable time frame. So considering all these aspects, the solubility and dissolution data can be demonstrated, and BCS3 waiver can be feasible if the solubility at pH 1.6 fasted state simulated gastric fluid is used. Solubility exceeds target concentration by many-fold for drug substance and drug substance complex. Mass balance is demonstrated for drug substance complex for suitable time frame. Here, mass balance is a critical aspect because it not only includes the parent but also gives, along with the degradants, it gives the confidence that the solubility is not overestimated. In fact, the 10% limit is in the guidance because to ensure that the solubility demonstrated is not overestimated. So mass balance shows that the drug substance, whatever the measured solubility, is not overestimated. And dissolution at the pH 1.6 for this general property of this drug product might result in greater than 85% dissolution for reference standard within 15 minutes. Coming to the final and tying scientific approach, that is PBPK for BE evaluation. The impact of drug degradation on in vivo performance of the drug product is evaluated by PBPK. Dissolution data at faster state simulated gastric fluid media and degradation data at multiple pH were incorporated into PBPK model. This PBPK approach provided the crucial scientific bridge between in vitro observation and in vivo clinical relevance, demonstrating that despite exceeding traditional degradation thresholds, the clinical impact was negligible. Overall, BCS Class III based Biowaiver is feasible. I have final question on this case study. Based on the case study discussed, which factor was most critical in overcoming the 10% degradation challenge, more than 10% degradation challenge for BCS3 waiver approval? A, adequate solubility study duration. B, PBPK modeling demonstrating no impact on bioequivalence. C, validated stability indicating analytical methods. D, totality of evidence including all of the above. I'll just give you a moment to think on this question. If you have an option, you can type in the answer. So the answer is D, totality of evidence including all of the above. I'll take a moment to acknowledge all wonderful people helpful for this scientific leap. Dr. OB Director, Dr. Partha Roy, DB3 Management, and a wonderful collaboration with ORS team, and acknowledgement to BCS committee. Thank you for your attention, and I would like to introduce or welcome Dr. Fangu to continue this on the same topic. Thanks.
Thank you, Usha, for the introduction of the background of this cladribine tablet case. So now I will give the introduction on part 2 of our presentation, addressing degradation challenges in BCS Class III Biowaiver applications through PBPK modeling. Here is my disclaimer. So as you have known, that this case of cladribine tablets, it has the challenging with the acid instability challenges. And then we also understand how AGDD evaluated BE based on the totality of evidence for the cladribine tablets case study, as introduced by Dr. Usha Kajagada. And now I would like to focus for understanding the role of PBPK modeling in regulatory decision-making for the approval of cladribine tablet. So cladribine tablet is considered as a BCS3 drug, but it has the degradation of the drug substance or drug substance complex in acidic environment. This has been observed in the in vitro studies. Per IM9 guideline, we know that in cases where the drug substance is not stable with more than 10% degradation over the extent of solubility assessment, then the solubility cannot be adequately determined. And if there is a significant degradation, and then this also precludes the BCS high permeability classification. So it is challenging to determine whether this product is eligible for BCS-based biowaiver. However, and you have seen that we updated the cladribine tablet, the PSG, in August 2024, and incorporated this BCS3-based biowaiver option in the revised PSG. We take the totality of the evidence, and as mentioned by Dr. Usha Kajagada in her presentation, and also PBPK modeling play an important role to support that a BCS waiver is applicable for cladribine tablets despite its degradation.
So as we know, the pH solubility data showed significant degradation in acid buffers, especially at pH 1.2. And regarding the dissolution, we see very rapid dissolution with over 85% dissolved within the first 15 minutes at pH 4.5 and pH 6.8. However, at 0.1 N hydrochloride, it dissolves less than 85% of the labeled amount within the first 15 minutes, and we see the decreasing amounts of the observed percent at later time points in acidic medium. This include in the medium at pH 1.2 and also in fast-give medium. But there is a less degree of decreasing amount in fast-give medium. So here is the question, whether this degradation will impact the BE of cladribine tablet. And we actually use the risk assessment approaches. These approaches include the literature evidence from in vivo studies assessing the impact of gastric pH change on the rate and extent of absorption cladribine, which can evaluate the impact of gastric change with the co-administration of PPI, whether this will cause any change of the PK of cladribine tablet. And also include the comparison of cladribine degradation with see the extent and the rate, whether they are different between generic cladribine tablet and the Mavriclad 10 mg tablet, which is ROD, across the physiological relevant acidic pH range. And then PBPK modeling was used to assess the impact of drug degradation on in vivo performance of the drug product. So we first developed the cladribine PBPK disposition model using the IV infusion PK data to estimate disposition parameters. From this figure, you can see the model predicted well of the PK after in vitro infusion of 3 mg cladribine tablet. Cladribine, it's the IV infusion. So then we developed the cladribine PBPK absorption model. In this process, we explored the two approaches, how to incorporate the dissolution and the degradation data. Figure A shows the model prediction using degradation data as model input to account for cladribine degradation. And when total cladribine, this total cladribine means that include the cladribine itself and the degradants in vitro dissolution data in fast-give medium. And the figure B shows the model prediction using cladribine only in vitro dissolution data in fast-give medium as model input without degradation data. Both of these approaches can predict PK well. And in addition, we compared incorporating the dissolution data at pH 1.2 versus incorporated the dissolution data at the fast-give medium. And we see that fast-give medium can be biopredictive for fasted conditions. So regarding the model validation, we use the PK data after oral administration of cladribine tablets with or without PPI. So for without PPI, as shown in figure A, actually we incorporate the in vitro dissolution data in fast-give medium. And for with PPI data, because you know like with PPI, there is an increase of the gastric pH, we incorporate the IVD, the in vitro dissolution data at pH 6.8. So the model replicates the results from in vivo findings, as you can see from figure A and the figure B. This shows that degradation at the lower pH does not have significant impact on PK for both ROD and the test products, as evidenced by the similar PK in the presence and absence of PPI. If the generic drug and the ROD, they have similar rate and degree of degradation. So further, for the model application, we conducted the VBE simulations using dissolution in fast-give medium, because we know fast-give is the biopredictive dissolution. And the VBE conducted between RLD and the test cladribine 10 mg tablets under fasting conditions, as you can see shown in the right figure. These simulations demonstrate the BE for RLD and the test cladribine 10 mg tablets. And when we incorporating degradation data and the biopredicted total cladribine dissolution data, this degradation data is expressed as % of degradation per hour at acidic condition. And then if it's BE, then the impact of degradation is similar on RLD and the test product. So based on all of these evidence, we know that there is a low risk of when there is a similar degradation rate of the generic drug and RLD. So in summary, a PBPK model was developed to assess the impact of cladribine degradation under acidic conditions on BE by incorporating biopredicted dissolution data, which is FASTGIF medium, as direct model input. And the model was able to predict the result from in vivo BA study that evaluated the impact of gastric pH changes on cladribine PK after the administration of PPI. In this case, it's pantoprazole. And the model was also used to demonstrate the cladribine degradation in acidic conditions with similar rate between generic product and RLD does not impact the BE of generic cladribine 10 mg tablet to RLD. So taking all of this evidence, the PSG for cladribine oral tablets was updated to include a BCS Class III based Biowaiver option. So I also have a challenge questions for you to consider and our better understanding how we incorporate dissolution profiles into PBPK model. For cladribine case, what dissolution profiles could be incorporated into PBPK modeling to better predict the impact of cladribine degradation on BE? A, total cladribine in vitro dissolution data in FASTGIF media, together with degradation data to account for cladribine degradation. B, using cladribine-only in vitro dissolution data in FASTGIF media as input without degradation data. C, in vitro dissolution data using FASTGIF medium. D, all of the above. Then the answer is all of the above. With that, I would like to acknowledge all of my colleagues listed here from Division of Quantitative Methods of Modeling from ORS, Aridam and Sharon for their work on the modeling part, and Dr. Lucy Fang, Andrew, Liang, and Rob and Lei for their guide, and also the reviewers and for the wonderful collaboration from OB, Office of Bioequivalence. Thank you so much for your attention. Next, I would like to welcome Dr. Min-Chu Cheng from Office of Bioequivalence for deliver her presentation.
Hi everyone, my name is Min-Chu Cheng. Today, Dr. Austin and I will present the topic on using modeling and simulation to correct carryover for long half-life drug products with incomplete washout. And here's the disclaimer. So this presentation only reflects the views of the authors, and it should not be interpreted as the views of the FDA. In our presentation, there will be two parts. In part one, we will first look at FDA current recommendations on BE studies for immediate release drug products with long half-lives and recommendations on carryover effect. And then we will discuss BE approaches and challenges in case studies with carryover effect. In part two, which will be presented by Dr. Austin, and she will talk about how we use population pharmacokinetic modeling to address incomplete washout scenarios. And she will also look at regulatory considerations on this model-based approach. Lastly, she will discuss opportunities to engage with FDA when facing similar challenges. So now let's dive into our presentation. Long half-life drug products are those with elimination half-lives longer than 24 hours. In general, randomized single-dose crossover design with an adequate washout period is recommended. And randomized parallel study design may be considered. Also, truncated AUC can be used to replace AUC 0-T for the extent of absorption comparison. And this can mitigate clinical challenges for prolonged sampling durations and follow-up. Carryover effects are residual effects of a treatment that can affect subjects in the following study period. So when there is an inadequate washout period, PK parameters may be overestimated and type 1 error rate may be affected. So all these issues can impact on the final BE determination. The two FDA guidances, first one is BE study with PK endpoints guidance for ANDA submission. The other one is recently published ICH M13A BE guidance for immediate release dosage forms. Both guidances have recommendations on BE study design. So the treatment period should be separated by an adequate washout period, and it's recommended to be at least 5 elimination half-lives. In terms of pre-dose concentration evaluation, when a pre-dose concentration is greater than 5% of the corresponding Cmax value, the guidance recommends to exclude the data from BE analysis. On the other hand, when the pre-dose concentration is less than or equal to 5% of the corresponding Cmax, the data can be included without adjustment. As I mentioned previously, a suitable truncated AUC can be used for drug products with long half-lives. The proposed truncated AUC should cover complete absorption phase of the drug. So now let's look at our case studies with carryover effect. In our case study 1, the drug product is an oral immediate release tablet, and B studies are designed as single-dose two-way crossover with a washout period more than 5 times of the reported half-life. And truncated AUC and Cmax are used for B evaluation. And in this case, there are approximately 60 to 80% of the subjects who completed the study having pre-dose concentrations greater than corresponding Cmax value in period 2. Also, there is an imbalance of carryover effect observed between the test and reference products. So after the exclusion, the total number of subjects included in the primary statistical analysis is less than 12, which is considered inadequate. Therefore, the BE results in this case are unacceptable and a new BE study is required. So in this case, a new BE study with a longer washout period is required. Some challenges may be encountered, such as the overall study timeline will be extended, and this may increase difficulties in subject recruitment, and a possible higher dropout rate and incomplete data collection. In our case study 2, the drug product is an oral immediate release tablet. So similar to case study 1, BE studies are designed as single-dose two-way crossover with washout period more than 5 times of the reported half-life. Truncated AUC and Cmax are used in this case. So there are more than 90% of the subjects enrolled in the BE studies having a significant pre-dose concentrations in period 2. So based on the guidance recommendation, if we remove all affected subjects from B analysis, this will result in a lack of data for B comparison. However, in this case, we are able to use a model-based approach to address this observed carryover in period 2. And Dr. Austin will discuss this case further in part 2. So when carryover occurs despite having an adequate washout period, we can see that period 2 data are compromised in crossover study, and traditional SCA is insufficient. And if we remove all affected subjects from analysis, there will be insufficient data for BE comparison. And in that case, if BE failed, we would not know whether the failure was due to actual product differences or it was due to data limitation. And the impact on applicants, it could be study failures, which means resubmissions and delays in drug product development. And now I'm going to hand over to Dr. Austin, and she will give you more insights on this model integrated evidence approach. Thank you.
Thank you, Dr. Chong, for providing the background. Good afternoon, everyone. This is Denise Austin from DQMM. In this part of presentations, we are going to cover how population PK modeling in the context of integrated evidence-based approach can be used in the scenarios where we experience carryover effect despite extended washout period, and how it can support bioequivalence assessment by preserving the data integrity. A robust PK model can provide scientifically sound alternatives to mitigate carryover issue. To describe it at very high levels, once we have the robust PK model, the individual parameter estimates of PK model can be used to simulate remaining concentrations, meaning carryover concentrations from period 1 at individual level through the end of period 2. Then we can use these concentrations to subtract from period 2 observed concentrations for all corresponding sampling time points through the conclusion of period 2 in order to obtain adjusted drug concentrations. This way, instead of excluding subjects with pre-dose concentrations greater than 5% of corresponding Cmax, we can preserve the data set. It will mitigate the chances of falling low in power, and therefore it can save the bioequivalence study if the study were to pass with sufficient power. The advantages of MIE-based approach can be better highlighted if it's compared to scenarios without implementing model-based approach. For the simplicity purposes throughout this presentation, we can call this approach as baseline adjustment approach. In the absence of model-based approach, the observed pre-dose concentration should be subtracted from all post-dose concentrations in period 2. With this method, the same fixed value throughout the entirety of period 2 will be deducted from observed concentrations, while in reality, the actual carryover amount is decreasing over time. Population PK modeling, on the other hand, can provide model-predicted carryover concentrations, which are otherwise unobservable in period 2, and therefore can capture declining carryover effect. This in turn means more accurate concentration adjustment and therefore more accurate bioequivalence assessment. To illustrate these two approaches versus each other, we can take a look at this slide. This is a very generic presentation of the scenario. The orange dashed line represents the model predicted concentrations, which is otherwise unobservable. What we can see in the real data set is the blue line concentration in period 2. As you can see, with model-based approach, the observed concentrations in period 2 can be corrected for declining carryover. Whereas in baseline adjustment approach, only the same fixed value, here denoted as C0, which is concentrations at pre-dose time point, can be deducted from all observed concentrations in entirety of period 2. Here we would like to share a success story of MIE-based approach in how it preserved the study integrity, how it helped recover the valuable data of a crossover bioequivalence study, where a washout period of more than 7 half-life was practiced. Regardless, all subjects showed carryover effect of more than 5% of Cmax. This is an example to demonstrate at very high levels how model-based approach was implemented, where to start with, what does it take to ensure model reliability, and how model-based approach was successful in accounting for carryover effect. This is an example that can set precedent for similar scenarios in the future. Starting with exploratory analysis, here when we plotted data on log scale, we identified that period 2 concentrations started at non-zero values, which is indicative of carryover effect. Of course, exploratory analysis is something that can be tailored based on your case, based on your data set. You can be creative in sets of exploratory analysis that you need to perform depending on the context that you're working on. Once we have the exploratory analysis, we can start model building. Here we used NonMem as the software, and we used first-order conditional estimation algorithm. We had data from both fasting and fed study, and since the data PK profile had very similar trend, therefore we pulled fasting and fed data despite some exposure differences, but this is another layer that can be decided based on your data set. Once you have your data set and you develop the model, you have your fixed effect parameters. Here we had 2 compartment model with linear elimination that describe the data the best. And after having the structural model, you can describe variability on relevant PK parameters. In order to describe further the variability, you can perform covariate analysis. Here we tested the effect of food on absorption-related parameter, and we found it significant on duration parameter for depot compartment. And eventually to describe, to account for formulation differences, we introduced treatment effect on absorption rate constant. In addition to general goodness of fit diagnostic, which is not shown in this presentation because they are quite generic, here, given the pre-dose concentration in period 2, as is the main issue, the predictive performance of the model in terminal elimination phase is really crucial. And therefore, additional geometric mean plus for IPRET, which is model predictions at individual levels, versus observed concentrations, DV, were insulated specifically at period 2 pre-dose time point to further investigate the model reliability in predicting carryover. In this slide, we have an exemplary individual goodness of fit plot where you can see that the model predictions at both levels, population and individual levels, are in close alignment with observation, which are shown in points, gray points here. In the next couple of slides, you see the PK profile, concentration time profile before adjustment for carryover effect. Here, the observed concentrations are shown with green points with 95% confidence interval as shaded area. This is before adjustment. And this is the same PK profile adjusted for carryover effect based on model predictions. And this is a superimposed version of the same PK profile showing before and after adjustment for carryover using PK modeling. And here we can see, based on 90% confidence interval for geometric mean ratios, that using IPRET-based corrected concentrations, bioequivalence was assessed and was concluded successfully. To ensure successful implementation of MIE approach, 2 key elements are essentials. The first one is robust model building, and the second is comprehensive model validation. For robust model building, we need three components: model building using sufficient clinical data, appropriate structural model, which also brings the context of identifiability, and separately covariate inclusion. And we also have to adequately characterize variability in terms of intersubject variability on relevant PK parameters, and also the random variability as residual variability in your model. As for comprehensive model validation, this can be done through several approaches. It can be through goodness of fit, GOF evaluation. It can be through visual predictive checks. It can be through quantitative predictive checks. It can be GOF at individual level, validation that modeling approach produces results comparable to conventional methods when carryover is not present, sensitivity analysis, and demonstrated ability to detect differences between test and reference formulations. The presented case study here demonstrated how the MIE-based approach can account for carryover effect while preserving the dataset integrity and mitigating the risk of underpowering that would occur if we had to exclude data in the absence of modeling. It also demonstrates that MIE-based approach are proven pathways to successful ANDA approval. And the key to success is to engage early with FDA. You don't need to wait until you encounter problems. We encourage you to discuss your modeling strategies during model development planning through MIE pilot program. This program is a dedicated platform that goes beyond the standard meeting formats and is intended for complex modeling discussions. The example scenarios where you can engage with FDA via MIE pilot program are provided in this slide for your reference. It can be common modeling issues across multiple products. It can be complex modeling approaches for non-complex products. And before closing, I would like to provide some key considerations for preparing an effective MIE meeting request package. You should ensure that your proposal is scientifically justifiable and your questions are relevant, critical, and clear. You should provide sufficient documentation of your proposed modeling approach, and that can be how your proposed approach can address the question of interest. Sufficient details for underlying assumptions and model building process. Clear model verification strategies supported by data. Risk analysis assessment, and finally, model files and supporting dataset. There are some resources here for your further references if needs be. And with that, I can close the presentation and pass it to the next presenter, who is Dr. Karen Lee. Thank you very much for your attention.
Thank you, Dr. Austin. And good afternoon, I'm Karen Lee, and I'm joined by my colleague, Dr. Duan Nguyen, and we are from the Division of Therapeutic Performance 2 within the Office of Research and Standards at AGDD. And today we will be discussing updates and the current landscape for study population selection and additional safety mitigation strategies in bioequivalence studies. And this is our disclaimer that this presentation reflects the views of the presenters and should not be construed to represent FDA's views or policies. The purpose of our presentation today focuses on the specific considerations related to subject safety in pharmacokinetic bioequivalence or PKBE studies, and we will cover three main topics today. First, we will discuss the reassessment of patient-based product-specific guidances, or PSGs. Then moving on to the additional features to ensure subject safety in healthy subject-based studies, specifically on pharmacokinetic or PGX considerations, and the use of concomitant medications, specifically on antiemetics as a safety mitigation strategy. And we will begin by exploring how to reassess recommendations for patient-based PSGs. To start off, I would like to reference the recently published guidance for industry on M13A bioequivalence for immediate release solid oral dosage forms. The guidance states that to reduce variability not related to differences between drug products, studies should normally be performed in healthy subjects unless the drug carries safety concerns that would make this approach unethical. In addition, if the investigated active substance is known to have adverse effects and the pharmacological effects are considered unacceptable for healthy subjects, the study may be conducted in the targeted population instead. However, I would like to comment that conducting a patient-based studies may have some inherent challenges, including recruitment difficulties, especially in the case of rare disease indications, potentially a higher cost and a longer study duration, and as well as the intrinsic variability due to the underlying differences in condition among patients. And lastly, the guidance further highlights that if the highest strength of a drug product cannot be administered to healthy subjects for safety or tolerability reasons, a single dose study in healthy subjects using a lower strength may be acceptable. In the PhD development process, we utilize a comprehensive evaluation framework for study population selection, which includes several important assessment factors. And this includes non-clinical toxicology profile or preclinical safety data, safety data in patients and healthy subjects if available from new drug applications, abbreviated new drug applications, post-marketing data, and public reports such as literature. We also evaluate the drug exposure as well as the associated safety risk at the dose that's intended for BE studies. We also compare the relevant safety profiles of other drugs within the same class. And lastly, pharmacological possibility with regard to whether the safety risk is expected based on the drug's mechanism of action. This slide highlights some of the core and common safety criteria to guide study population selection for PKBE studies. However, I want to note that this is not an inclusive list, as each individual drug have their own specific warnings, precautions, and contraindications. And some of the criteria include cytotoxicity, which involve direct cellular damage, genotoxicity and carcinogenicity, as with regard to the potential for primary malignancy, reproductive toxicity with regard to embryo-fetal toxicity and fertility impairment. And other warning precautions and contraindications in the labeling include hepatotoxicity, cardiovascular related toxicity, hypersensitivity, blood dyscrasia, effects on the central nervous system. And for each of the potential adverse effects, there are some additional consideration factors, including the severity, reversibility, dose relationship, and the time onset of each potential adverse event to determine the appropriate population for the PKBE study. In healthy subject-based studies, to ensure the subject safety for this population, we have adopted several mitigation strategies. These are listed on this table. The first one is to reduce exposure to healthy subjects, either using a lower strength as a BE study dose or using a parallel design to limit the exposure for giving subject to a single dose as opposed to 2 doses in a crossover design. And other mitigation strategy include having specific exclusion criteria. In this case, the example is to exclude subjects with abnormal liver function tests. And associated targeted monitoring include to monitor for liver function tests. And in the case of reproductive toxicity, we can also consider restricting the population to only those not of reproductive potential. And also in a case where there is a higher risk for adverse events in the geriatric population, we can also consider excluding this population as well. And the last 2 factors, which are the main topic for today's presentation, the first one is pharmacogenomic or PGX factors. If the drug is metabolized by a polymorphic enzyme that could lead to a higher exposure in certain genotype, we can consider excluding these subjects due to safety or PK reasons. And for the last topic, which is concomitant medications, in the case of GI adverse events, we may consider the use of antiemetics as a mitigation strategy. And over the next few slides, I want to highlight some examples for the reassessment process. So the first drug of interest is available at 2 strengths with indication-specific recommended dosage in patients. And with regard to the PK, it increases proportionally with increasing dose. And the major relevant safety concern that trigger the initial patient-based recommendation is due to the severe liver injury that occur in patients following short-term exposure and also cardiac failure that were observed in patients. And therefore, based on these considerations, the original PSG recommended a steady state crossover study in patients who are already receiving a stable dose of the drug. And as additional safety data in healthy subjects accumulate from NDA and ANDA programs since the initial PSG publication, it prompted a reassessment of this PSG of the drug of interest. And based on the emerging data available in healthy subjects, it has supported a potential revision to a healthy subject-based studies. And the majority of the adverse events in healthy subjects were mild to moderate in severity, and they were usually manageable without any pharmacological interventions. And additional analysis also revealed that the incidence and the severity of hepatic and cardiac abnormality in healthy subjects were higher at the higher strength comparing to those who received the lower strength, which indicate that there is a potential drug exposure response relationship for the safety risk of interest. And based on the available information and the emerging new data, I mentioned that a revision to a healthy subject-based study using a lower strength with appropriate monitoring may be a feasible option. And moving on to example 2, this drug is available at 3 strengths, and the recommended dosage in patients is using the middle strength once daily for 7 days and titrate up to the highest strength once daily. In this case as well, the PK is dose proportional in the range that cover the middle and highest strength. The major relevant safety concern is the specific toxicity with early onset, which require patients to use a dose titration scheme. In healthy subjects, serious adverse events were also observed. And similarly, the original PSG recommendation recommended a steady state crossover study in patients who are receiving a stable dose at the highest strength. Upon reassessment, the incidence of this specific toxicity with early onset was dose-related and occurred within 7 days of initiation in patients. And based on the pre-approval data in healthy subjects, it was observed that the incidence of AEs of the potential toxicity in subjects who receive a single dose of the highest strength was 4-fold higher comparing to those who receive the middle strength or similar sample size. And all AEs of the potential toxicity in subjects who receive single doses of the middle strength was mild and manageable with non-pharmacological interventions. And importantly to note that there were no cases were reported in real-world settings in patients who followed the approved titration regimen as stated in the labeling. And based on this data, it was also determined in this case that a revision to a healthy subject-based study was an appropriate option. I would like to note that in general, a revision to a healthy subject-based study is often considered a minor revision, given that a study conducted in patients following the previous recommendation is still considered acceptable. However, in healthy subject studies, the product will be determined whether it's a high-risk or non-high-risk product, and additional study may be needed depending on the risk assessment based on the M13A guidance, as opposed to a typical single study conducted in patients following the standard of care. And now I will move on to the second topic, which the integration of PGX considerations in BE studies to ensure subject safety. And I want to cite the M13A guidance, which states that phenotyping and or genotyping of subjects may be considered for safety or PK reason. The integration of PGX information into BE studies may serve several important functions beyond the typical health or safety screening that usually occur prior to study or during the screening period. In terms of safety, it allows us to identify and exclude subjects at higher risk for adverse events due to the genetic variation in drug metabolism. It can also help us to mitigate exposure-dependent or severe adverse events. With regard to the study design, it may optimize crossover study design for drugs with long half-life by ensuring an adequate washout period and also to manage inter-subject variability. And lastly, for drugs with long half-life, it may be helpful to ensure balanced group allocations in a parallel study design. So currently, there are over 390 drugs counted per API that contain PGX information in their respective FDA labeling. However, as of September 2025, there are only 17 published PSGs that include PGX-related recommendations. And the common enzymes are listed on the slide, including the cytochrome P450 enzymes, including CYP2D6, CYP2C19, and 2C9. And the non-CYP enzymes include G6PD, DPD, TPMT, and NUDT15. When considering the integration of PGX into the PSGs, we consider several factors, and this includes how the polymorphic enzyme affect drug pharmacokinetics and the resulting variability between individuals, whether the genetic variation have any impact on the safety differences across genotypic subgroups, and whether the labeling carries any contraindication or requirement for testing of the polymorphic enzyme prior to initiating treatment in patients. And lastly, the implication of PGX on both clinical and pharmacokinetics in healthy subject-based studies with regard to safety or study design-related considerations. And with that being said, I want to illustrate a specific example with Simponimod. So Simponimod, which is a sphingosine 1-phosphate receptor modulator indicated for the treatment of relapsing forms of multiple sclerosis, and it is available in 3 strengths. The labeling recommends testing patients to determine the CYP2C9 genotype prior to treatment initiation, and also avoid use in patients with a CYP2C9*3*3 genotype as a contraindication. And lastly, the labeling also recommends to reduce the maintenance dose in patients with certain specific genotype. And notably, the CYP2C9 genotype have a significant impact on Simponimod PK, with AUC increased by approximately 61% to 285% in subjects who carry 1 or 2 copies of the CYP3 allele. And also, the mean half-life is also prolonged in a specific genotype. The current PSG recommendation for Simponimod includes additional comments to exclude subjects with CYP2C9*3 allele, as those listed on the slide. And the rationale for such inclusion was due to the up to 4-fold higher exposure in CYP2C9*3 allele carriers. The labeling carries a contraindication and dose adjustment for specific CYP2C9*3 genotype. And also, from the study design perspective, is to minimize carryover effects following a crossover study, or to ensure a balanced group of subjects in a parallel study due to the long half-life. And with that being said, with the relatively limited implementation of PGX in current PSGs, with only 17 PSGs, this represents an area of opportunity to enhance subject safety through PGX approach in BE studies. And next, I will hand it over to Dr. Wynn to discuss the concomitant medications in BE studies.
Thank you, Dr. Lee. So now that Dr. Lee has discussed the recent changes in patient-based PSG to healthy subject-based PSG, as well as the importance of considering PGX information in BE studies, I will wrap up the presentation with the last piece of additional feature to ensure subject safety in BE studies, which is concomitant antiemetics, and then wrap up our presentation. So why do concomitant antiemetics matter in BE studies, and especially in healthy subjects? Gastrointestinal or GI events, such as nausea and vomiting, are among the most commonly reported adverse reactions in BE studies conducted in healthy subjects,
And from the safety perspective, this can significantly affect the subject tolerability as well as compliance or retention within the study. And then from the PK results perspective, vomiting within a specific time frame can impact absorption of the oral drug products and increase the variability in PKs, which potentially require removal of subjects from PK analysis to avoid confounding the BE conclusions, which is a recommendation in our general PK/BE guidance. For these reasons, mitigation strategies for vomiting in BE studies for high emetogenic drugs may be crucial to subject retention and sample size estimation. Although generally concomitant medication is prohibited in BE studies in healthy subjects, concomitant antiemetics in these cases may be considered provided that its use does not compromise study integrity or affect BE outcomes. Next, I'd like to discuss the current state on the use of antiemetics in PK/BE studies, as well as a few of our recommended considerations for its use. Currently, there is a limited number of PSGs, which are 5 published PSGs covering 3 active pharmaceutical ingredients that include antiemetic recommendations in the PSG. Based on our internal FDA research, we found that most of the corresponding BE studies that were submitted for these products, which were over 90 BE studies, incorporated the antiemetics that were recommended. Based on the research outcomes, we also found the following considerations that should be considered when antiemetics are being implemented in BE studies. First, we recommend that the antiemetics being selected should have low risk of PK interactions with the study drug, and this can be either via absorption, elimination, or GI motility. Next, we also recommend consideration of use of non-oral routes of administration for the antiemetics being selected, and this may include intramuscular, intravenous, or transdermal. And lastly, we recommend that any antiemetic being considered, and as well as the administration method, should be specified in the study protocol with the rationales. Lastly, before closing out the presentation, I'd like to discuss a recent example in which the PSG was revised to reduce the study dose as well as addition of the antiemetics in the PSG. The drug being discussed is Selectapac oral tablet, and the reference listed drug is available at 8 different strengths ranging from 0.2 milligram to 1.6 milligram. It is a prostacyclin receptor agonist indicated for the treatment of pulmonary arterial hypertension, WHO Group 1, to delay disease progression as well as hospitalization. The labeling recommends a recommended starting dose of 0.2 milligram given twice daily, and then the dose can be increased in increments of 0.2 milligram twice daily, usually at weekly intervals, to the highest tolerated dose, up to 1.6 milligram twice daily. If a patient reaches a dose that cannot be tolerated, it is recommended that the dose is reduced to the previously tolerated dose. The original PSG was published in 2016 that recommended a fasting and a fed single-dose PK/BE studies using the 1.6 milligram strength, which again is the highest available strength for this drug product. After the PSG was published, there were several pilot BE studies that were submitted to the agency via multiple channels, including controlled responses that showed a withdrawal rate due to vomiting up to 100% in these studies that were conducted in healthy subjects. Following these new submitted data, the PSG was revised in 2020 to recommend a fasting and a fed single-dose PK/BE study that used 0.4 milligram strength, which is the the next to lowest strength. And the revised PSG also include a recommendation to include treatment for nausea and vomiting in the study protocol. Following the PSG revision, the pivotal BE studies that were submitted since then for approved ANDAs showed a reduced withdrawal rate due to vomiting at up to 43.8%, which is still relatively high compared to other submitted BE studies. However, this is way more feasible as compared to the previously submitted pilot BE studies at the highest available strength. Unrelatedly, the PSG was also revised recently in 2024 to remove the Fed PKB study pursuant to the M13A guidance, but the study dose remained at 0.4 milligram strength with additional comment regarding treatment for nausea and vomiting. With that being said, I'd like to summarize the content that has been discussed by Dr. Lee as well as myself. Clinical assessment for the selection of BE study population and dose is based on a combination of non-clinical toxicology data as well as clinical safety data that are available from NDAs, ANDAs, post-marketing data, as well as literature, drug exposure profiles, and pharmacological plausibility. Patient-based PSGs undergo continuous reevaluation based on cumulative safety data as well as evolving regulatory experience with a specific drug product. The development of new PSGs as well as the revision of existing PSGs can incorporate emerging safety mitigation strategies for healthy subjects, including, as Dr. Lee discussed, pharmacogenetic factors, as well as use of non-high strength, as well as concomitant antiemetic therapy, as I just discussed. Lastly, strategic study designs incorporating appropriate measures may be implemented to ensure subject safety without compromising regulatory decision-making. Here's our acknowledgement slides. We would like to acknowledge DTP2 colleagues as well as leadership, Dr. Shan, Dr. Tran, Dr. Bay, Dr. So, and Dr. Kim. ORS colleague, ORS leadership, Dr. Lineberger, Dr. Zhang, as well as other collaborators within OGD, including the Generic Drug Development Safety Committee and colleagues within Office of Safety and Clinical Evaluation. Thank you. I'd like to hand it over to Dr. Gong for her presentation.
Thanks for the kind introduction. So this is Yu-Ching Gong from the Division of Quantitative Methods and Modeling, ORS OGD. Today, I will present a collaborative work between us and Office of Biostatistics regarding the method to extrapolate of pharmacokinetics bioequivalence to an alternative comparator when the case, the R&D and RS is not available. And our biostats colleague Wanjie will also join me in the panel discussion to answer questions related to this talk. So in my talk, I will discuss this alternative approach for establishing pharmacokinetics bioequivalence, the PKBE, when the reference listed drug or the reference standard R&D and RS is unavailable. And I will analyze case studies that demonstrate the application of using an alternative comparator in BE studies. And I will do a step-by-step calculation through case demonstration to show how we calculate bioequivalence with this alternative approach. So first, I'd like to give you a review of our general practice. So for most drugs, the focus of bioequivalence is on the release of the drug substance from the drug product into the systemic circulation. So during such BE studies, an applicant compares the systemic exposure profile of a test product to that of the R&D designated in FDA's Orange Book. And this reference standard in the in vivo B comparison, ordinarily it's the R&D. And if FDA cannot select the R&D as the RS, for example, in the case where the R&D is withdrawn for reasons other than safety or efficacy, FDA may designate one of the generic as the RS. However, there were cases when the R&D is discontinued, but there's no generic approved, so there's no RS available. So under these cases, if an applicant wanted to develop a generic referencing this R&D, it's no direct way for them to compare the in vivo bioequivalence against the R&D. So here I'm introducing an alternative approach that extrapolate bioequivalence using an alternative comparator, which the generic applicant can conduct a relative bioavailability study with this alternative comparator and establish bioequivalence based on comparing the results with the historical R&D data. So this alternative comparator can be a product with different dosage forms for the same route of administration. And by leveraging publicly available bridging studies, such as the study conducted by the R&D holder during the NDA submission, they can take together the information here, and then this is an alternative pathway to guide the generic applicant who is interested in developing product when no R&D and RS is available in the market for them to perform the in vivo comparison. And I'll go to more details in my following slides. So this slide shows a schematic representation of this approach. In the middle part, I insert a yellow box for the alternative comparator C. So in this approach, we have two sets of data. One is the historical data, where I have the comparison for my R&D and this alternative comparator C. And the second set of data is the new PK study, where the generic applicant compare its test product versus this alternative comparator C. So in this approach, instead of the direct comparison to the test versus R, so we leverage this alternative comparator. That means we're comparing the relative ratio for the R&D versus alternative comparator, and for the test versus this alternative comparator, by taking these two sets of data, then to extrapolate the 90% confidence interval of the T by R ratio, and eventually we demonstrate bioequivalence by comparing this 90% confidence interval against our standard criteria, 80 to 125%. So here is my first case study. The case 1 is an oral suspension where the R&D and RS is not available. And this R&D was approved as a new drug application through the 505(b)(2) pathway. And the NDA holder demonstrated its oral suspension is bioequivalent to the oral tablet product. And in the review, it indicates the 90% confidence interval fall within 80 to 125%. So based on BE transitivity, demonstrating bioequivalence between the test product, the T, and the oral tablet formulation, serving as the alternative comparator C here, will establish BE transitivity across all three products, the R, the T, and the C in general. And I know many audience may be familiar with BE transitivity, as this is a well-established approach commonly applied when the generic RS is used for the in vivo BE comparison in the cases where the R&D has been discontinued from the market. So for our research, we focused more on the cases where this R&D and this alternative comparator is not equivalent, because under these cases, the BE transitivity cannot be directly applied. So let's go to my next case, my case number 2. So this is for an extended release oral tablet. Similarly, the R&D and RS is not available. And this case is related to a recently published product-specific guidance. The product-specific guidance for trazodone hydrochloride extended release oral tablet. The guidance was just released last week, October 1st. So for this product, the trazodone extended release oral tablet was approved as an NDA through the 505(b)(2) pathway. And the NDA holder performed a relative BA study that compared its ER tablet to the immediate release tablet, the IR tablet. So in this case, the BE transitivity cannot be directly applied, as this reference drug, the R&D, or it's not BE to this alternative comparator, the IR tablet. However, the 90% confidence interval for T by R can still be extrapolated by leveraging this data versus the new study data that contains the T versus its alternative comparator, and ultimately can determine the BE between the T and R. And I will go to the detail about this method. So this is the available historical data for trazodone ER tablet. So during the NDA submission, the NDA holder performed a two-way crossover PK study that they compared a single dose of the ER tablet versus the IR tablet 3 times a day. So here I put a figure here, showed how this study, the profile concentration look like. So they follow the same total daily dosage for the ER tablet versus the IR tablet 3 times a day. So as I showed in the summary statistical analysis of the PK parameter to compare the IR versus ER in the table here, you can see the bioequivalence between the ER tablet and the IR tablet 3 times a day was not demonstrated, as the GMR for Cmax is 2.5 and the GMR for AUC is 1.24. However, with this fundamental information available, so a generic company can perform a similar study as showed in the study 1, that historical data. So I'm showing study 2 here on the right side of the slides. So the generic company can perform a new study that compares its test product versus this IR tablet 3 times a day, following similar study design as it was conducted in this NDA study. So this alternative approach utilized the relative difference between these two dosage form, the ER versus IR, so eventually calculating the 90% confidence interval for the T by R kind of extrapolate based on the relative ratio when they compare to the IR tablet. So in an intuitive way, you can think about like the ratio I just showed for AUC when it compares the IR versus the ER is 1.24. So you are expecting a similar ratio when you compare your test product versus its IR, because you expect your test product behave similarly as the R&D when it compares to this alternative comparator IR tablet. So here I'm putting some details about the statistical method here, and you can find the complete equation in the draft guidance on trazodone hydrochloride that was recommended on October 1st. So for this approach, first, we can calculate an unbiased point estimator of the log transform geometric mean ratio for the T by R that can be calculated based on the equation listed on the screen here. And this equation leverages the estimate of log transform geometric mean ratio from study 1 and study 2 that includes the comparison from T versus the comparator and R versus the comparator. And regarding calculating the 90% confidence interval, so first, in the guidance, we recommend to perform an F-test, which is to test the equality of the intra-subject variance between this study 1 and study 2, and then calculate the 90% confidence interval either on the homogeneous or heterogeneous assumption of the variance. So here I took a snapshot of the equation that it's calculating 90% confidence interval under the homogeneous variance assumption. And in the product-specific guidance, you can find the complete equation, including the other scenario where you need to calculate confidence interval under the heterogeneous variance assumption. So this is depending on the results of the F-test. So I also wanted to show you a step-by-step calculation, how the bioequivalence can be concluded. So in this case, I created a hypothetical case example for my study 2. So my study 2 is the test product compared against this alternative comparator C. So in this hypothetical case example, I assume my test product behave the same as the R&D when it compares against this alternative comparator. So I assume my GMR is the same and my 90% confidence interval is the same. For this case, you can notice that for the study 2, my alternative comparator became to the denominator. So in this case, when I create this hypothetical case example, I just flip the GMR. So here I'm using the example for trazodone, the data for AUC, where in my study 1, when I compare the IR versus the R&D, the C by R, my GMR is 1.24. Here for my study 2, the hypothetical case example, I just flip the GMR to 0.81. Similarly, I flipped the 90% confidence interval to create this hypothetical new study for this case demonstration. And I keep the same N number for the number of subjects. So N1, it's the number of subjects in sequence 1. So in my sequence 1, I have 11 subjects. In my sequence 2, I have 12 subjects. And on the right side for the blue box here, I also put the log transformed theta value. So for my study 1, my log transformed theta is 0.2116. And for my study 2, it's a negative 0.2116. And I also calculated the mean squared error, MSE, based on my GMR and confidence interval and my N number that is showing here. So my first step is to calculate the geometric mean ratio for my T by R, leveraging the study 1 and study 2 data. So I use the equation that is listed here. Basically, I took the theta, the log scale theta from study 1 and study 2 and add them together to get my T by R ratio. So my T by R ratio calculated from here on log scale, it's the theta, it's 0, and on original scale, the theta equals to 1. So this is what I expected because in this hypothetical case, I created a case where I assume my test product behave the same as my reference product when it compares to my alternative comparator. So my T by R ratio equals to 1 here. And then I tested the equality of intra-subject varies between this study 1 and study 2. As I previously showed, we do an F-test to see if the intra-subject varies between the two studies is homogeneous or heterogeneous. So in this case, I took my mean squared error from both studies, and then I calculate the 95% confidence interval for these two values, follow the equation that is projected here. And then I find my 95% confidence interval for these two values, it's 0.4152 to 2.4086. And because this confidence interval contains 1, so we consider the values of two populations from study 1 and study 2 are equal. And then based on this, I follow the equation that is listed on the product-specific guidance under the homogeneous assumption, where I use the mean squared error as the pooled mean squared error. Then I calculate my 90% confidence interval following the equation here with the assumption that my variance is homogeneous under the pool variance. Following the equation here, and I calculated my 90% confidence interval for my T by R. In this case, it's 88.64 to 112.81. This is on original scale, and I find this is within 80 to 125%. Then I concluded the bioequivalence between T and R is demonstrated in this case. So here is a summary of my talk. So through our internal research, we demonstrated an alternative approach for establishing bioequivalence through an alternative comparator. So this alternative comparator can be a product with a different dosage form for the same route of administration. And the 90% confidence interval for T by R will be extrapolated based on their relative PK ratio for both products compared to the alternative comparator. And the bioequivalence will be concluded if this extrapolated 90% confidence interval for the T by R ratio fall within the acceptance criteria. And our standard criteria is 80 to 125%. So this approach provides an alternative method to guide the generic applicant who is interested in developing product when R&D and RS is not available in the market. And I hope today's talk through this case demonstration, you understand this approach better. We strongly encourage applicants to engage in early communication with the FDA during your product development. For example, through the model integrated evidence industry meeting pilot, if you're considering this alternative approach for demonstrating bioequivalence, because we think for each product, you may have product-specific considerations for your specific product. So I think you saw these slides from the previous presentation, so I'm just putting here again regarding the model integrated evidence industry meeting pilot, because this is helpful to get specific scientific communications regarding the quantitative methods and modeling techniques. And this is applicable for complex modeling approaches for non-complex products, which is suitable for the case that I discussed here. And this will provide a dedicated regulatory platform for interactions with FDA regarding any quantitative method and modeling questions. So here I put my acknowledgement slides. This includes big group of people from OGD ORS and OGD Office of Bioequivalence and Office of Translational Science and Office of Biostatistics. So without these people, we cannot put this complete story here and put it into the PSG. So I'd like to thank everyone that is listed here and many more. And I'll be in the panel to answer questions, and also our biostats colleague Wenjie will also join the panel to answer questions related to this talk. Thank you.
Thank you so much, Dr. Gong, and to all of our Session 2 presenters for sharing their expertise and insights on novel bioequivalent study design recommendations. Your presentations have provided valuable guidance for advancing generic drug development. Let's now take a short afternoon break, and we will reconvene at 3:10 p.m. Eastern Time for the Session 2 panel discussion. During the break, please continue to submit your questions via email to orsevents@fda.hhs.gov with the subject line of AGDD 2025, and we will address as many questions as possible when we return. We'll see you all back in a few minutes.